Thank you so much for the opportunity to speak here today. As John said, my name is David Bianchi. I was recently a PhD student in the Luther Schulten Lab at the University of Illinois, and now I'm working as a research scientist in the National Center for Supercomputing Applications there as well. And what I'd like to talk to you about today is some recent work we did on functional characterization of the minimal cell JCBI Syn3A proteome. And so in the Luther Schulten group, what our main kind of modus operandi is cell simulations that integrate various types of experimental data, whether that be cryo-ET, some tomograms from the Vila group that you've seen previously, fluorescence microscopy or omics data, and integrate these into these whole cell kinetic simulations that we can then run on high performance computing facilities. And so Syn3A was a really intriguing prospect for us, and working with John and the synthetic biology group there for reasons that probably many of you are quite familiar with, the small size of the genome and the really well-characterized nature of the proteome in comparison to some really well-studied organisms like E. coli that we can see here on the right. And so with this in mind, recently we published a whole cell kinetic model for Syn3A in Cell earlier this year. And I got some really interesting results there regarding predictions of doubling times over a population, DNA content predictions, and also key metabolic pathways that are present in Syn3A. With really great data from our collaborators in the SENS group, providing lipidomics, as well as collaborators at JCBI. But one issue, especially for us making these types of whole cell models that we still face, and that's still a very interesting question moving forward, is these genes that have unknown function, or at least are relatively unannotated up to this point. And so this is something that we are really motivated to continue to dig into a little bit further and see if we can kind of clarify that picture a little bit more. And so with that in mind, recently we published this paper in the Journal of Physical Chemistry B, trying to computationally analyze many of these unannotated genes in Syn3A using various computational techniques. And so I'd like to talk about that work today that we did with John and Clyde at the JCBI, as well as James Pelletier, who spoke last week from the Centro Nacional de Biotecnología in Madrid. And so there's several previous groups that have analyzed these kind of genes of proteins, I should say, of unknown function in JCBI Syn3A using various techniques, studying protein-protein interaction, threading type techniques, and secondary structure prediction. But one kind of feature that wasn't... Hello?
David, I'm very sorry, but we don't see your slides going on. So...
Oh, I'm sorry.
Yeah, you seem to be
stuck.
It's
staying on the first slide still. I'm sorry. I did not notice that. My apologies. Thank you. Thank you for interrupting there. Let me try again from here. Now is it let's see. Let me try just sharing my entire screen then. Okay, I will just try sharing the entire screen instead of just PowerPoint. That will probably be a better way here. So now can you see the slides moving?
Yeah.
Okay, excellent. I'm so sorry about that. Thank you for letting me know. Perhaps I can just start from where I was, and hopefully the introduction was still somewhat cogent. Do you think that's a good avenue to proceed by, John?
Yes, I think we're good.
Okay, excellent. So as I was saying, we recently had published this paper in Journal of Physical Chemistry B, looking at some of these proteins of unknown function, and working with John and Clyde and James. And so there's been previous groups over the last few years that have looked computationally at trying to get at functional annotation of some of these proteins via various techniques. But one kind of feature that wasn't present at this time, and that's been mentioned earlier in work talked about today, whether by David Goodsell or Christoph as well, as being quite useful, was kind of the advent of AlphaFold in protein structure prediction. And probably I don't need to convince you too much of the efficacy of this methodology. And so what we really wanted to do was use an individualized approach with these about 90 or so proteins in Syn3A that had unknown function, that included techniques like AlphaFold, as well considering the genomic context in which these genes are in Syn3A and comparing that to other bacterial organisms, as well as in conjunction with sequence alignment and other literature and interactomics type data that's available in SynWiki that Christoph had described to us earlier. And so our kind of general idea here was doing these types of bioinformatic analyses on these 91 or so proteins of unknown function, and then trying to get a more molecular level insight for these specific proteins so that they could be implemented into our kinetic whole cell models, so that these models could be refined and continually made more robust, so we can get some predictions on the effects of those individual genes on whole scale cell dynamics that can be then kind of interrogated experimentally with our collaborators. And so I'll give you a few kind of vignettes of things that we looked at, but one protein in specific is associated with this idea of the FTSC filament and kind of its role in setting perhaps the midplane of a cell in a cell division period. And so the gene that we specifically had been able to kind of provide an annotation upgrade for was this locus tag 239, which we feel that we can annotate as a EZRA-like protein. We have our AlphaFold structure prediction in blue in subpanel A there. That's aligning quite well by structural alignment techniques to the experimentally determined structure from subtilists. And something that David Goodsell had shown really excellently in those hydrophobicity plots that he had earlier in his presentation is we can see the portion of the structure that would be embedded in the membrane here is quite hydrophobic in red there, and as well as having some support in panel D here from the genomic architecture in subtilists being quite similar to what it is in Syn3A in terms of the gene products neighboring this gene coding for EZRA. And so another complex of interest that's quite important in many cells and in Syn3A is no different, is the ATP synthase. And in the mycoplasma especially, this is very important in maintaining the pH gradient across the membrane, actually pumping proteins out of the cell. And so we have this large macromolecular complex here with so many protein components. And all of these that I'm showing you here on the right in B subtilists are present as well in Syn3A in consecutive order in the genome. But one thing that hadn't been characterized yet was the gene immediately preceding all of these ATP synthase genes. And we, again, using AlphaFold and examining the genome architecture, we're able to assign this functionality to the regulatory protein ATPI that's involved in modulating the efficiency of the complexation of the entire ATP synthase machinery at the membrane. And so kind of in conclusion of this much larger work that was done, we were able to provide upgraded functional annotations for dozens of proteins that were previously of unannotated or unclear function in Syn3A, including proteins involved in cell growth and division, pH maintenance, and other important processes. And similar to what Christoph had also mentioned and is available through SynWiki, we were able to provide structural predictions for all of the protein coding genes via AlphaFold, as well as a visualization and analysis notebook that you can interrogate each of these predictions with. And so included in the supplemental data of this paper, we have spreadsheets giving each of our predictions for each of the genes, as well as this database where you can visualize all of the AlphaFold predictions. And in addition, Christoph and Tiago and Jorg have included many of these main predictions already in AlphaFold in SynWiki here. And so Danke, and thank you again to them for really providing this wonderful resource. And not only for Syn3A, but for Subtilis as well. SubDWiki is an amazing resource. And just some kind of an additional tool, I think, that can be useful in addition to those predictions available on SynWiki is we provided this notebook where you can interrogate each of these protein coding genes and choose any of the various models for a specific protein generated by Syn3A, and then visualize it with associated error coloring, chain coloring, kind of whatever type of visualization we choose. And we also have associated with this 250 gigabyte database of the sequence and genetic data that's used by AlphaFold to generate these predictions. And so I'd like to thank all of our coworkers on this project, my advisor, Sanluthi Shulton, John, James, and Clyde, and my group members, Zane, Troy, and Ben, who are also really instrumental in bringing this project to life. And so thank you so much for your attention, and I'd be happy to take any questions.
David, really nice work. Thank you for including me and Clyde and James with your Illinois group. So I mentioned earlier the idea about moonlighting, and James Daubenspec from UAB pointed out that in their view that a large number of mycoplasma proteins moonlight. So as you look at the AlphaFold data, do you see, so have you thought about looking at it in the sense that it may be predicting proteins that it may make two predictions that seem contradictory but perhaps aren't?
Yeah,
and
no, this is a really
how would we go about trying to sort all of that out?
Definitely. And that's a really interesting question. I know I've talked with James Daubenspec about this before and several of the other collaborators. And I think that's something that was nice in terms of those associated genetic data that we have with these AlphaFold predictions is you can get at least some sequence-based support, possibly in terms of seeing if there's any similar substructure in a protein, similar structural modules to proteins that do maybe a different type of function that you would expect. And as well, I think that will be something that can be enhanced by interactomics studies, kind of deeper studies, like what Christoph had mentioned for other mycoplasma species that are available in SynWiki. But from the computational perspective, that's something in this study we weren't able to interrogate in a really deep way yet, I would say.
I certainly think as we get more interactome data and maybe more of the kind of imaging data that George was showing us today, if you see proteins hanging out in two different areas, or you see evidence of membraneless organelles, that there are concentrations different places. But this is, I think, AlphaFold is a remarkable tool that is really changing all of biology. But I'm betting that there are clues there about moonlighting. It's just hard to pick them out.
Definitely.
Do you see predictions that disagree with what we have been calling things?
Disagree. Let me think. I guess at one point, one example that I can mention, but this was in a kind of very earlier iteration of the assembly. I think it maybe actually was in the SYN1 assembly possible, that ATPI gene I had seen characterized sometimes as some type of sugar transport associated gene. And I think there was some substructure motifs that supported that. But as we kind of looked deeper and took into account all these different facets, including the genome organization, I think it became more clear to us that this would be ATPI type functionality there. But I can't think of too many examples where it disagreed. I think more in general, it was just being able to kind of upgrade these annotations and maybe even understand functionality in a deeper way. Another example I could think of off the top of my head that I know you're familiar with, John, is one of the genes that was discussed in James's Pelletier's cell paper that might be involved with LSU large subunit assembly was as well something we're able to upgrade the annotation of in this paper, and that seems to at least hold in a general sense as why that might be affecting changes in morphology between SYN 3.0 and SYN 3A that are seen.